Therefore the name, but we'll not get confused in much detail. We'll only remember this, that it's basically matures as well as originates inside the bone marrow. Okay, this much will be sufficient. You do not go for all these trivial informations. So what exactly happens? B lymphocytes initiates humoral immune response. Now what does it mean by humoral? Humoral immune response means they are the thing which will produce these antibodies. Okay. So B lymphocytes are responsible for humoral immune response, which also initiates the production of antibody. We will come to those points, the typical these things, that how exactly this antibody formation takes place and all this in coming days. And so this group of immune response is called humoral immune response, where B lymphocytes will look after the production of antibody. So antibody mediated immune response is nothing but humoral immune response. Now what about cell mediated immune response? Cell mediated immune response is activated when there are some typical antigens, not any, only two major antigens, which are either viral antigens, mainly viral antigens, or cancer antigens, cancerous antigens, like shall I say CA125. Now due to a GI cancer, an individual will have a increased production of CA125 or something like in hepatic cancers. They'll have alpha fetoprotein and things like that. Now these antigens, whenever are present in the body, cell mediated immune response will try to eliminate the cancer with a preferential rate. Okay, so it has been seen in recent research also, that viral antigens and cancerous antigens are preferentially eliminated by cell mediated immune response, which is taken care by whom? T lymphocytes. Remember, so T lymphocytes will look after cell mediated immune response, whereas B lymphocytes are responsible for humoral immune response. तो cell mediated immune response में जो है आपका natural killer cell, then cytotoxic T lymphocytes, I'll come to those points, are mainly the players. Okay, but in B lymphocyte mediated, antibody mediated, what we call immune response, antibodies are the main players. We'll come to those things. So humoral immune response is mainly initiated in case of where your antigen is what? Bacterial antigen. ठीक है, जैसे कि for example you are taking a vaccine against like BCG you are taking. Okay, now in that case what will happen? There is an increased probability that anti-BCG antibody will be very, very high. Okay, but this is a very typical case. But when you are taking this thing, polio vaccine, or like shall I say a COVID vaccine, there is a probability that your memory T cells are increasing. I'm not saying that it will not, the titer of antibody, anti-COVID antibody will be very, very less. But typically in a classical scenario, the antiviral vaccines are basically increasing the population of memory T lymphocytes. Okay, whereas antibacterial vaccines, okay, BCG, DTP and so on, they will increase the number of memory B lymphocytes. Okay, now memory cells are what? Memory cells are those kind of cells which will enhance the immune response during the real case of an infection. Okay, so those things also we'll discuss. So these are the things. Okay, one more thing that we want to discuss today itself is this. So what exactly happens is whenever the B lymphocyte is produced in the bone marrow, they'll get released, isn't it? There will be like the spongy bones, like this it will be if we draw it. So consider that this is a spongy bone. Okay, like tibia or femur or humerus, things like this. Okay, so they'll get released in the bone marrow through the spongy outgrowth. Okay, so B lymphocytes has been released. Now these B lymphocytes are not primed. Okay, these are virgin, so they have not encountered any kind of antigen till now. So on the surface of the B lymphocyte, we know that there are 5 different types of antibodies, isn't it? Let us remember all over again. IgG, then what? IgA, then what? IgM, IgD, and then IgE. Okay, if you remember like this. Now in a normal human being, okay, in a normal human being, highest amount of antibody without any infection, a normal person is there, he'll which antibody will be highest in the blood? IgG. Then which one? IgA. Okay, then which one? IgM. Then which one? IgD. Then which one? I'm from low highest concentration to lowest concentration in a normal. For example, you have isolated the serum of a patient, not patient but a person who's not having any kind of immunological issue as such, and you are doing a chromatography in order to separate which are the different classes of antibody present in the person who is a normal person without any infection, everything is okay. So in that case you will see that concentration of IgG will be highest, then IgA, IgM, IgD, IgE. Okay. Now if then B lymphocyte is unprimed, so which type of antibody is this? Consider that it's an unprimed what you call unprimed means virgin. Okay, not yet encountered any kind of antigen. In this case it will be IgM. Remember this. Okay, so on the surface, many a times on the surface of an unprimed B lymphocyte there will be IgM. IgG will also be there, but in the this thing it will be IgM. So what happens is it's an unprimed B lymphocyte. It will come and in the circulation and it will look for some kind of foreign particle. Okay. Now whenever a foreign particle has been introduced inside on the surface of the what you call B lymphocyte, now B lymphocyte will start behaving like an antigen presenting cell. Okay. Now what is that antigen presenting cell? APC. All the macrophages mainly are what we call APC, antigen presenting cell. Okay. Now B lymphocyte is typical because it can also have its main function is to produce antibody, that is fine, but it can also behave like a what? Antigen presenting cell. Okay. The unprimed B lymphocyte can act as an antigen presenting cell and eventually they can identify the foreign antigen and then initiate immune response against it. Now it's very paradoxical that whenever a bacterial antigen will enter into the system, then B lymphocyte will be primed or B lymphocytes will be activated. Now if there is a viral antigen, let us talk about this thing, COVID vaccine. Now in vaccine what happens, many a times it can happen that a hand-in-hand reaction takes place, B lymphocyte, T lymphocyte simultaneously. But if it's a true this thing, what you call unmodified virus, let us think like this, that you have inactivated the virus but not made it into a subunit vaccine. Now in subunit vaccine what happens, the whole part of the virus is not there, only a small spike protein or things like that. But if it is a sabin or salk vaccine, so what will happen, the whole structure of the polio is there. So whenever you are putting it into the circulation, what will happen is this thing will start, what you call cell mediated immune response will start. Okay, but since the oral vaccines are what you call inserted through the oral route, there is a local this thing immunity which is taking place in the gut itself. But in blood, if you inject a full scale virus, so what will happen is you will see that cell mediated immune response has been initiated with a preferential rate. Okay, so I have summarized it from a book, the whole, shall I say, the overview of immune response, how exactly it takes place. Okay, so I will be sharing with you this. Let us save this.